Okay, welcome to our first video in a series of 4 on glycolysis. So we're going to start taking a look in detail about some of the more important metabolic pathways, and we are going to focus first on energy metabolism. So the glycolysis metabolic pathway is found in almost all cells in all life forms, so it's incredibly important, and we're going to essentially break the glucose and harness the energy. As we look at our goals, overall, we're going to talk about the 10 steps of glycolysis, but really today, let's say we're going to summarize. We're going to look at the big picture in this first video. And we're going to differentiate between 2 phases where we have to invest energy and where we generate energy. All right, this is a picture of many, not all, of the various cycles in human metabolism. And today we're going to focus in, glycolysis sits right up here. So let's blow up that section and let's take a look at what we're looking at. So in glycolysis, we're going to go from glucose to pyruvate. And we'll also see in a little bit later video that we can get to glucose from many different points, right? We could start with starch, lactose, galactose, glycogen. So glucose is incredibly important to us, so our bodies have several different routes to get to it. Okay, let's look at a big overview of glucose metabolism. It is 10 steps, okay, and each of those steps is enzymatically controlled, and we'll eventually get to pyruvate, which is the end point from glycolysis. So pyruvate is a alpha keto acid. So think about that term. We've got our carboxylic acid. Let's see, that's what I want. We've got our carboxylic acid. We have our alpha carbon, and we have our ketone group on that alpha carbon. So we have an alpha keto acid. It's this small metabolite, this 3-carbon molecule. Okay, and it has many different fates depending on cell type, cell conditions, right? We can take it and change it into acetyl-CoA to go into our citric acid cycle. We can form lactate. So we'll talk about all those later, but many different fates of that once we make it. All right, let's look at a big overview here of the glycolysis piece. So a few things. Energy comes from a redox reaction. This occurs in the cytosol of the cell, and as we already said, converts glucose to pyruvate. There are 10 reactions. The first 5 we consider our energy investment, and the second 5 we're going to give off. We're generating energy. So when we look over here on the right in blue, for each glucose we net in these first 5 steps, we don't net, we use up 2 ATP for each glucose, so it takes energy. At the end of those first 5 steps, we end up splitting that glucose molecule into 2 smaller molecules that feed into the energy generation phase, highlighted in yellow here. During that phase, we make 4 ATP, 2 NADH, and 2 pyruvate. So if we come down here to where we show our net, notice that we went from glucose to 2 pyruvates. We made some energy right now, 2 ATP, that we made right during glycolysis. But we also make 2 NADH that carry electrons down to our electron transport chain and generate more ATP at the end of that chain. So that's a future video. Okay, so glycolysis, by the way, lysis, to split, so we end up splitting that glucose up. All right, remember that each of these NADH carry 2 electrons, so we're actually carrying 4 electrons into our electron transport chain. All right, this is a great slide. I really like it because it's a great overview. So let's take a look at everything we have, right? This black text right here is telling you the, right here, right, is the name of our reactants and products. Notice that if I am looking at this, so in my first reaction, I'm forming glucose 6-phosphate, and that glucose 6-phosphate becomes the reactant for my next reaction. Okay, so we have this chain. The pink name over here is the name of the enzyme that catalyzes each step. So these steps would be really, really slow. They might happen without the enzyme, but not in any sort of speed that would work for our bodies. The black text over here is telling what is happening, right? So, for example, in step 1, we're going to have a phosphorylation. Okay, we see it in the black text. And the enzyme that catalyzes that is hexokinase. Remember that term kinase, where we're taking a phosphate from ATP and adding it onto something else, transferring it. Notice over here we have our co-substrates. So in the first one, that co-substrate would be the ATP. All right, let's take a look just at one. Oh, and then, of course, way over here, we have our structures. So as an example, let's just look at step 1. So we start with glucose. We're shown the glucose molecule here. We know that this is going to be a phosphorylation catalyzed by hexokinase. We're going to use an ATP in this process. And we know that phosphorylation, we're going to add a phosphate group. So when I look here, we get glucose 6-phosphate. And here's that phosphate added on right up here. So this is actually an important step. So glucose enters the cell through this glucose transporter, and once it's phosphorylated, that transporter can no longer recognize it and transport it out. So essentially this step traps that glucose in the cell. All right, we'll talk about all of these steps in detail in the next video. For now, let's skip ahead to the energy generation phase. Oh, real quick, look up here. As we come down through this phase, we end up with 2 of these smaller 3-carbon molecules, okay? Glyceraldehyde 3-phosphate. So as we move into the energy generation phase, each of these reactions is essentially happening twice, right? I have 2 of these going to 2 of those to 2 of those, and so on and so forth. Okay, we will see as we come down here, we're getting ATP out during that energy generation phase. All right, last thing I want to, I meant to take those slides out and I evidently did not download the right slides. So let me just tell you what I was going to talk about next. And we'll look at these 2 slides in our next video. All right, 2 terms that I wanted to get in here today. One is the committed step. All right, this is the first irreversible step. First irreversible step in a metabolic pathway that it's irreversible, at least under physiological conditions, okay? After which molecules are committed, and I'm going to talk about, so we don't want to define a term using the same term, to the pathway. All right, so for example, if we had some substance. And enzyme A converts it to another substance. And that substance maybe has a couple options, right? Enzyme B could convert it to a new substance, or, you know what, enzyme Z could convert it to a different substance. And then we could keep going. We're just going to keep our pathway going. And finally, maybe this is our intended product. In this metabolic cycle, this step right here would be my committed step. All right, because after that, and we're assuming that that's an irreversible step, but after that step, there's no other outlet for this molecule, right? There's no other branching. Once it goes through this and becomes this molecule, its only path is to eventually turn into this intended product. All right, back here, this first step wouldn't be the committed step because this product, number 2, could go either way. It has options. But now the committed step for synthesizing product 6 here would be this one that's controlled by what I listed as enzyme C. All right, so that's the committed step. Let's talk regulatory step. All right, regulatory steps are ones in which the enzymes respond to cell conditions to speed up or slow down reactions. So enzymes respond to cell conditions, which could be the presence of certain metabolites or certain, I should use the term, certain other biomolecules. Could be pH, it could be lots of different conditions, but respond to cell conditions to speed up or slow down process. All right, we will look at these in the next few videos and talk about what is the committed step in glycolysis. What are the regulatory steps? All right, that is it for video 1.